everybody. Welcome back to the second half of today's lecture and the last lecture of this particular module. And last time we left off with telophase 1, which begins the prophase of the very next cell division. It's prophase 2. And just like with prophase 1, your nucleus dissolves and your chromosomes begin to align towards the middle, your spindle fibers attach to the centromeres, and you wind up in meiosis 2 at the end, where all of your chromosomes are lined up. Now, unlike meiosis 1, in meiosis 2, your chromosomes, your sister chromatids, are pulled apart. And which one goes to the left cell, so to speak, and which one goes to the right, happens independent every time it happens. So you get your genetic variation from crossing over during prophase 1, and you now get genetic variation during meiosis 2 and into anaphase and telophase 2, because your chromosomes, the daughter sister chromatids, randomly assort into one of the two cells, and they do so with no relation to any of the other chromosomes. It's called independent assortment. So you don't know really what's going to happen until it happens. And so you move through metaphase 2 into anaphase, and instead of an entire chromosome moving to a daughter cell, it's just a sister chromatid that's moving towards one half of your cell. And then finally in telophase 2, and eventually then again in cytokinesis at the very, very end, you recreate a nucleus, and cells begin to differentiate either into a single oocyte, where one egg is produced, or a spermatozoa, where 4 sperm cells are produced. And so obviously one is male, one is female. That depends on what type of cell is produced, what type of gamete is produced. And again, meiosis provides a tremendous amount of variation, because when it comes to crossing over, you can shuffle genetic information just for one person in a really large amount. And if you were taking AP chemistry, I would make you memorize exact figures here. But the idea is you can shuffle the deck a whole lot of ways. And then after the deck is shuffled, you can split the cards into different stacks for daughter cells, a whole bunch of different types of ways as well. And so you can have millions of children and never have a twin naturally, right? Just by chance. It's very unlikely that any couple who lived any amount of time would ever have an identical twin just by chance. Okay, I think I'm going to lecture to that. Now, it's important to note that many plants undergo sexual reproduction, and they can have a very odd doubling up of chromosomes, or they can have a life cycle that it's a gametophyte life cycle and a sporophyte life cycle. So some plants can have an entire organism that's just in, and then they can have a different organism that's 2N. So not all organisms, like us humans, have a very short reproductive cycle where the gametes exist as a cell, and then they become a diploid cell immediately after fertilization. Some organisms can exist as a haploid cell for quite a long time before they undergo sexual reproduction. Now, one of the last topics we're going to talk about in this particular section is something called a telomere. And telomeres are little caps on the ends of your chromosomes that have a protective function, and they also seem to act a little bit like a biological clock. And every time a cell divides, the telomeres get a little bit shorter until eventually, when the telomere gets short enough, a cell will not divide anymore. It'll undergo apoptosis instead. Now, a karyotype is what a scientist would use when attempting to study the structures of your chromosomes, how healthy your chromosomes are, and whether or not you have the appropriate number of each of your chromosomes. Okay, so One way to figure out what gender your child is going to be is to take a little bit of the amniotic fluid and perform a karyotype, and you can see its chromosomes line up and see maybe an X and a Y chromosome, and you know it would be a boy, or an X and an X chromosome, and you would know it's a girl. And it happens occasionally that when gametes are being produced during your metaphase and anaphase 1 or metaphase and anaphase 2, something known as non-disjunction can occur. And this is where chromosomes or sister chromatids do not properly disconnect from each other. And you wind up with either too many cells, or too many chromosomes, and that's called polyploidy, or too few chromosomes, and that's called aneuploidy. And generally speaking, these lead to some pretty bad genetic disorders. So, for example, if you have 3 chromosomes on your 21st chromosome, you get Down syndrome or trisomy 21. And if you get 2 X's and a Y, you can get Tay-Sachs syndrome. And we'll go through a few of these disorders from the textbook that you can read about, but they can have varying degrees of severity, from somewhat mild to extremely life-threatening. There are some situations where if you get the wrong set of chromosomes doubled up, or you don't have enough chromosomes, you have 1 instead of 2, or maybe 0, then as an unborn child, a spontaneous death will occur and the child will be lost. Okay, I think I've already talked about sex chromosomes to the degree that I think I want to. Oh yes, the book does talk about Turner's syndrome, which have only 1 sex chromosome. And people can survive with Turner's syndrome, but they generally have to get hormone therapy and treatments so that they can have a somewhat normal life. Okay, so the last 2 topics we're going to talk about are cellular differentiation and stem cells. So apart from gametes and apart from regular cell division, that G0 phase that I talked about can happen, and that's cellular differentiation, where a cell leaves a generic cell life cycle and becomes a neuron, or becomes a lung cell, or becomes a muscle cell, and it does some specialized function until it dies. Well, a stem cell is a very valuable type of cell because it can reproduce multiple times and then become some kind of specialized cell, right? So when you're a very young child, you don't have all the brain cells that you need. And so you have what are called embryonic stem cells. And they can divide hundreds or thousands of times to grow your brain, or to grow your heart, or to grow your kidneys, or your liver, or any of your other organs. And they can specialize out into them. And so doctors really like to play around with stem cells because they hope to be able to cure some disorders. So instead of having a kidney transplant, for example, maybe they could just grow a new kidney inside of you from stem cells. That's kind of one of their goals. And there's 2 types of stem cells. Again, there's the embryonic stem cells, which occur naturally. And then there's adult stem cells, which happen by what's called induced pluripotency. I don't think you need to know that particular term, but that's the process of taking an adult stem cell and making it divide more times than it normally would. Now, adults do also have stem cells that occur naturally, like bone marrow. A bone marrow inside your bones is an undifferentiated stem cell that eventually becomes a red blood cell. And your marrow turns from a reddish color when you're young to a yellowish color when you're white, or when you're old. Yellowish color when you're old and gray, you get white hair. And that's because all of your stem cells get replaced with fat cells in your bones. and your bones get rather brittle and unhealthy as you get older because of this. So the point is that those stem cells in your bones, your bone marrow, becomes red blood cells. And one of the things that scientists have done, again, is to take those kind of cells and induce pluripotency and induce the ability to divide more than just into red blood cells, right? Because the adult stem cells, they have already divided many, many times, and they're near the end of their ability to divide more. And so to be able to use adult stem cells, you have to do some reprogramming to get them to divide more. So back to our focus question, and then off we go. What are the stages of meiosis, and how does meiosis provide genetic variation? Well, hopefully I have given you enough terms and ideas to explain this question. If not, do please ask me. I want to make sure you know it. Otherwise, thank you so much for listening.